So welcome everyone to this new session of Cafés Sistemáticos. Today the talk is going to be in English. I'm happy to introduce João Farminhão, who is a postdoc and lecturer at the Free University of Brussels. He did his PhD at this same institution on systematics of Afrotropical orchids, which is the topic he's going to talk about today. So the talk is going to be about 30 minutes, and then we will have time for questions, which can be in either English or Spanish, because João, even if he's going to give his talk in English, he can understand Spanish very well. So yeah, welcome, João, and it's all yours.
Thank you, Mario, and thank you to the Sociedad Española de Botánica for this opportunity for me to talk today. So without further ado, let's talk about Mr. Darwin, about, technical problem, about Mr. Darwin, about orchids, and about Hawkmoths, and specifically about Darwin's famous prediction on the pollinator of this spectacular orchid from Madagascar, Angrecum sesquipedale. And I'm going to introduce also this pollination syndrome, sphingophily, this fancy word for hawkmoth pollination. But before moving on to the topic of today, I would like to briefly present you what I've been doing as research in the past 6 years during my PhD. So I've been studying the systematics of angrecoid orchids, and here I present you an overview of the floral and vegetative diversity of this group of epiphytic orchids. So here you can see Angrecum sesquipedale, so this is the main character of today's talk, but it's only one of 800 species approximately that are mostly distributed in tropical Africa and Madagascar, Afrotropics. And they are renowned for their great chromosome, diversity of chromosome numbers. So this is unparalleled among all orchids. But today we are going to focus on pollination biology. So in this talk, in the first part, I'm going to present an overview of the history of Darwin's famous prediction on the pollinator of Angrecum sesquipedale. And for the second part of this presentation, I'm going to present an overview of the natural history of sphingophily in the Afrotropics, namely its frequency, spatial patterns, newly identified morphological patterns, and finally, I will finish with an evolutionary time frame, an intro to the evolutionary time frame of this syndrome. So, as you know, the supposed co-evolution between Angrecum sesquipedale and these giant moths from Madagascar is commonly used in teaching ecology and evolution. And this figure here, for example, is extracted from the most widely used textbook on evolution, Futuima. But unfortunately, I must tell you, as an angrecoid taxonomist, that the plant represented here is not Angrecum sesquipedale. Well, it's a little bit sad, but now you know this is another species of Angrecum. And I leave to my colleague Simon Verlan, who is the specialist on this genus, to tell you exactly what species it is. Now, this moment in the history of science is so widely known, is so part of our common imaginary biological folklore, that there's even a book for children telling the history of this discovery and how it took 130 years between Darwin's hypothesis and the first confirmation of pollination by these giant hawkmoths. So if you want to get your sciencey kids excellent gift, you may introduce them to this book, which you can also see presented by a science educator on YouTube. But now let's do it more formally. So with the chronology of the events. So we now have to go back to 1862, when exactly 1 month after Christmas, in January 1862, Mr. Darwin did some epic unboxing. So he received a shipment of exotic orchids, namely the famous Angrecum sesquipedale. And he was immediately struck by the length of the spur of this orchid that attained 29 centimeters long. So on the very first day he received the orchids, he wrote to his friend Joseph Dalton Hooker a letter in which he wrote this famous sentence: "Good heavens, what insect can suck it?" And it took only 5 days in another letter to Joseph Dalton Hooker to advance with his prediction. What proboscis must the moth that sucks it must have? So this is the first written evidence of Darwin guessing the pollinator of Angrecum sesquipedale as a moth, a gigantic moth. And on the very same year, only 3 months later, Darwin included this extraordinary orchid in his seminal work on the pollination of orchids. And this prediction was immediately met with a lot of skepticism. So Darwin was ridiculed for this hypothesis. How would it be possible an insect to have proboscis that long? But a few years later, Wallace, Darwin's admirer and fellow naturalist, wrote a paper in defense of Darwin's hypothesis. So here on the right-hand side of the slide, you can see a plate that accompanied his defense of Darwin's hypothesis. And this is how he pictured the possible pollinator of Angrecum sesquipedale. And it is interesting to read his paper, very good writing skills, as he compares the future discovery of the pollinator to the discovery of the planet Neptune. Because I don't know if you know or you recall that the planet Neptune was discovered in the 19th century after its mathematical prediction. So there was a French scientist, Urbain Le Verrier, that accurately predicted Neptune's existence and its position. And this was in the imaginary of scientists in the 19th century. And Wallace compared these, and they said that naturalists that would look for it would be equally successful as astronomers looked for Neptune in the skies. Now, just as a curiosity, let's go to 1873, when a teenager, 19-year-old William Alexander Forbes, wrote to no other than Nature a short letter in which he says that the pollinator is probably of Angrecum sesquipedalum is probably a hawkmoth. So this is the first written connection between Darwin's orchid and hawkmoths. And it is now picturesque for us scientists in the 21st century to see that what kind of papers were published in Nature that times. Those were simple times. Well, now we have to go back to the beginning of the 20th century to see the discovery of the candidate pollinator of Angrecum sesquipedale, which was found, I'm sorry for these technical issues, by these two entomologists who described it as a subspecies predicta, the predicted one, of this moth that is also found in tropical Africa. And here you can see its extraordinary long tongue. But it was only at the end of the 20th century that there was confirmation of this interaction in Madagascar. First in a flight tent in Madagascar. You can see here the first ever pictures taken in August 1992. And they were later studied in the greenhouse in Germany at Erlangen. And this historical greenhouse, unfortunately, was demolished. So only photos are left. And these discoveries were made by Professor Lutz Thilo Wasserthal, that you can see here. Now, adding a little bit to this story, last year, the taxonomic status of Xanthopan was clarified. And apparently, the species, the moths in Madagascar, correspond to a different species from Africa. So now we recognize two gigantic moth species in tropical Africa, Morgani from mainland Africa and Praedicta from Madagascar. And you can see here the extremely long coiled tongue of these species. And here I give you the reference for the full story of this scientific discovery since Darwin's prediction. Now, I would like to draw your attention to the difference in average proboscis length between Madagascar and mainland Africa. Moths have quite longer proboscis in Madagascar, and we may wonder what effects this has in the local floras. So now let's move on to these questions, general questions about sphingophily, hawkmoth pollination in Afrotropics, starting by asking ourselves, how frequent is this syndrome? Before answering the question, a short historical overview of the study of hawkmoths in Africa. So everything started with Stefan Vogel, who at the beginning of the first half of the 20th century studied the flora of tropical Africa and identified the list of characters that we now associate with hawkmoth pollination more generally. Also, I would like to draw your attention to these graphs here on the left-hand side of the graph of the slide. So here you have in black the distribution of proboscis length in moth species, and in red the distribution of nectar pollen distances, tube length in flowers. And as you can see, there are some distribution peaks, and these peaks have been associated with different size classes of hawkmoths. So basically, three phylogenetically defined groups of hawkmoths with different proboscis lengths. Now, I also make a plea for reading the classics. So this work by Vogel is extremely interesting to read. Unfortunately, I don't read German, and if you, like me, don't read German, you also have access to this seminal work in an English translation from 2012. And it's not only about Africa. Stefan Vogel also talks about some species we know well from our Iberian Peninsula. So namely, he talks about the pollination mechanisms of Kixia cirosa. Now, going back to sphingophiles, how do we recognize a sphingophile? So here I present you the list of characters. So the white flowers, some have dissected, so fringed margins. They are typically night flowering as a response to the pollinator's behavior. They are strongly scented, and I would like to draw your attention to this telltale long nectar pollen distance. So this is what we can look for, namely in herbarium specimens, in the description of floras, and go through the entire African flora to pinpoint the candidate, the probable sphingophile. And that's what I've been doing, starting in PhD. So I went through the description of approximately 40,000 species of the African flora. And here I show you what I found. So we have the distribution of hawkmoth length, proboscis length, co-plotted with the distribution of floral tube length, or more generally, nectar pollen distances. And as you can see, the previously identified pattern is found here as well. So different peaks in the distribution of sphingophiles. So this is only for probably hawkmoth pollinated species. And also a pattern that strikes the eye is the apparent excess of long-tubed flowers in Madagascar compared to Africa. And overall, these sphingophylls represent approximately 3% of the entire flora of Madagascar and approximately 2.3% in the flora of tropical Africa. What's also very interesting to realize is that angraecoids are the greatest component of hawkmoths, hawkmoth pollinated species in Madagascar, and this is not the case in tropical Africa, mainland tropical Africa. Now, to describe these differences, these different peak modes, distributional modes, I propose a classification scheme recognizing micro, macro, and mega sphingophilous species. And these are associated with pollination by the largest hawkmoths, in the case of mega sphingophylls, by large size but not that large sphingoid moths in the case of macrosphingophylls, and finally the microsphingophylls pollinated by macroglossine short-tongued hawkmoths. Here you have an overview of the diversity of micro, macro, and megasphingophilous species. So if we look at the most spectacular examples of megasphingophily, these represent 41 species in tropical Africa and 21 species in Madagascar. Of course, I cannot show you all the extraordinary plants in this guild, but I'm going to present some examples. So, as a biologist initially trained in Portugal, I'm very familiar with this tree, Melia azedarac, that is commonly found in parks, in streets, and in this family Meliaceae, there's this staminal tube that makes the distance between the pollen and the nectar reward. And we may wonder if there's potential for elongation of this staminal tube in Meliaceae. And here's the answer coming from Madagascar. So in Madagascar, there are some Meliaceae with quite long staminal tubes that go up to 12 centimeters. But if this doesn't impress you, let's upgrade, let's increase the size of these corolla tubes. Now, I show you one of the most evolutionarily distinctive species of the Malagasy flora. This monotypic genus, Tsuala, as it's pronounced in Malagasy, Tsuala tubiflora, which is a small shrub that presents these remarkable tubular flowers. Now, a third and last example, of course, orchids. I'm biased to talk about orchids, and they show really the most extraordinary examples of long tubes. So everyone knows about the Angvecunceski pedale, but I leave you with two other examples. So we have, for example, this terrestrial species, Cynorchis mammothus, so the mammoth Cynorchis, that goes up to 27.5 centimeters, or this undescribed species of Solenangis, that is being described by my colleagues from the Missouri Botanical Garden, that goes up to 33 centimeters. So in 2022, there's still undescribed diversity among these extraordinary guild of hawkmoth pollinated plants. Now, after having an idea of this diversity, we can wonder if there are any spatial patterns among sphingophylls, among hawkmoth pollinated species. Now, let's start with some geography, with some biogeography of tropical Africa. So here I show you a map with the different bioregions in the Afrotropics. And when I started to look more closely to the results, to the distribution of species, it appears that some areas, namely this coastal strip of East Africa, called the Swahilian coastal, is particularly rich in long-tubed flowers. And this remains to be clarified, what may be behind this pattern. Now, if we look at the ecology of these species, the habitats where we can find most sphingophiles, it appears that some patterns recur, some patterns appear. So sphingophiles look particularly abundant, namely in hilltops, in rocky hilltops, or in rocky outcrops more generally, or, for example, in marshlands, in wetlands. And what we can tentatively suggest is that, well, these habitats in general have quite patchy distributions. And we may wonder if there is a selective advantage of hawkmoth pollination in these cases, because these insects, these large hawkmoths, are excellent flyers, and they can possibly connect these quite disconnected populations in patchy habitats. This is just an hypothesis. Also, regarding the concentration of sphingophiles in rocky hilltops, we may wonder if hilltopping behaviors, so these concentration of individuals of insects during mating season, could also play a role in explaining these patterns. Just speculation for now. Now, to corroborate these spatial patterns, or to give you an example of these spatial patterns, I show you here the section Acidanthera of the genus Gladiolus. So in this section, all species are confined to rocky habitats, with one exception, Gladiolus iroensis, that grows only in temporary ponds. So this pattern appears evident in this strictly hawkmoth pollinated lineage. And there are other examples. Now, we all know about white flowers, strong jasmine-like scents, nocturnal flowering, but are there any additional sphingophile traits that still need to be pinpointed, identified? Well, I show you some results of a first overview. So, for example, among legumes, about 3 legumes, it is interesting to verify that independent lineages, different lineages of legumes have evolved large white flowers with a big yellow blotch on the uppermost part of the flower. And these yellow blotches can either be petals, staminodes, for example. And in the case of 7 different families, it is also interesting to verify that in these white flowers, dark red markings appear as well. And hawkmoths apparently are blind to dark red. So eventually, this increases the contrast of the floral display. And I forgot to tell you that apparently hawkmoths are attracted to yellow. So this would explain this pattern in legumes. More recently, I also realized that one species of probably hawkmoth pollinated orchid also has this possible yellow flag. Now, if we look at some angraecoid taxa that have particularly long spurs, and they probably share the same pollinator species, it is interesting to verify what mechanisms may act as reproductive barriers in these species that share this unique pollinator. And apparently, at least in these 2 lineages of angraecoids, there is a difference in column length, in gynostemium length, that may translate into divergent pollinaria attachment sites. And it's also interesting to verify that these pairs of sister species have adjacent distributions. So these may be an example of reinforcement associated with these shifts to long-term hawkmoth pollination. Now, finally, about the time frame when the sphingophily first evolved. So I collated all information about dated phylogenetic trees and I co-plotted it with the timeline of sphingophily evolution, Hawkmoth evolution. So it is interesting to note that this pollination syndrome is likely one of the youngest, one of the major youngest pollination syndromes. So starting only in the Cenozoic towards the Oligocene. And it is interesting if we now take a look at the species that have the longest spurs or the longest floral tubes. They are conditioned by the age for the appearance of the largest hawkmoths from the genus Xanthopan in the Miocene. If we focus on angraecoids, there are at least 3 independent transition shifts to this pollination by the longest proboscis hawkmoths. And these were dated at different times. Of course, we always have to take this with a grain of salt, these divergence time estimates. But in any case, they appeared at different times and they correspond to different pollinator shifts. And I'm going to finish with this question about coevolution, because we often talk about this perfect coevolution between the large hawkmoth and the large angraecum. So this is the scenario A, the Darwin's coevolutionary race. But in the case of angraecoids, we may have instead a case of a pollinator shift. So plants that increase their spurs in response to this available niche of hawkmoths that already had long hawkmoths, and these may have evolved with other species if we look at the dated phylogenies. So I would like to thank you for your attention. Muchas gracias. So here I show all the institutions with which I've been working, the funding agencies as well. And special thanks to Mario, Andrea, and my friend Felina Bensadi, who suggested me to talk here for Cafés Sistemáticos. Muchas gracias.
Thank you, João. That was a great talk, very exciting, and it's great to see how we now have much more information and a lot more background about this topic that Darwin and Wallace themselves started a long time ago. So we've got time for questions. As I said earlier, you may ask in English or in Spanish. We also have our chat. You may type your questions and then I will read them to João. If there are any questions, ¿alguna pregunta? If not, I can start, maybe.
Okay, go ahead. Yeah.
So yeah, my main question was about the last slide, about
Yes.
whether this interaction between angraecum and Xanthopan is a case of coevolutionary race or a pollinator shift, because this is the question that usually comes up when discussing this system, right? So do you know how specific the relationship is in Madagascar between Angraecum sesquipedale and Xanthopan predicta? I mean, we assume that the orchid is only pollinated by this moth, but is the orchid the only plant that the moth pollinates? Or not?
So it's quite, how do you say, an unbalanced relationship. So for Angraecum sesquipedalum, it only relies really on these species of hawkmoth. But Xanthopan predicta feeds on a large array of species, including other species with very long floral tubes. So if we go back to the overview of the single-file flora, so in Madagascar there are 21 species that have these extremely long nectar tubes. So there's this guild of species that rely on Xanthopan. But Xanthopan can also have access to nectar from all the other of the plants, basically. So it's quite an unbalanced relationship. Now, in terms of the question coevolution, pollination shift, here what's also important to have in mind is the time frame for evolution. Of course, we always have to be careful about these age estimates, but apparently xanthopan is older than at least most of these long-tubed flowers. And there's this theory that these proboscis may also have evolved as a response to avoid predation during feeding. So for angracoids, they are certainly younger, apparently, than hawkmoths, these hawkmoths. So in the case of angracoids, the most likely scenario is a pollinator shift, which may have also then triggered a coevolutionary relationship, or more more extreme phenotypic variation, but probably it started as a pollinator shift in the case of these orchids. I don't know if I addressed the...
Yeah. I think, that clearly, what you said clearly supports the idea that the long spur in Angrecum has evolved in response to the to the long tongue of the moth, and not the other way around, right? So that would support the pollinator shift. But it's also interesting what you said at the end, that maybe it started as a pollinator shift, but perhaps after that, now there is some kind of coevolutionary race going on, right? That would be an interesting scenario to test with experiments. So I don't know if Carmen and Vicky are going to ask a question.
Hey, hello. Thank you, João, for the presentation. Here in León, we are following and we have enjoyed a lot the talk. We were just wondering, maybe we missed something, but about this yellow color and the red color appearing in different families.
Yeah.
I'm not sure if you mentioned which will be or if you have any hypothesis about the presence of these colors, or if it's a conserved trait along the clades, or if it's just appearing in the specific taxon. So can you explain us a bit?
Yes. Okay. Thank you for your interesting question. So regarding the yellow flag pattern, as I call it, so I initially identified it in legumes. So I don't know what mechanisms are behind the expression of yellow color in legumes, or why it is particularly easy for legumes to make these yellow flags. So I don't know the answer, but what's interesting is that in these different examples that you see on the screen, they correspond to different lineages of legumes, where you have closely related species that do not have this yellow pattern, or they don't even have yellow flowers. But also more recently, I found this example of orchid. Well, it's just speculation, but it's interesting to note that an orchid also has apparently this pattern. Also, to answer your question, if we know, maybe I understood that you also wondered what's the connection between yellow and hawkmoths?
Yeah.
So apparently there's some experimental evidence, some behavioral studies that show that hawkmoths react to yellow markings or yellow areas in a flower. So these may be like a trait that optimizes pollinator attraction. In the case of the dark red patterns, it's somewhat different, because here it's almost like an anti-patterning, because apparently, based again on experimental evidence, hawkmoths are not sensitive to red. They cannot see red or purple. And so if we imagine ourselves as hawkmoths, like seeing these flowers, the white pattern, of course, is very evident, clear, but these dark red areas, they just disappear. So apparently these may increase contrast. So imagine this example. So the letter G here on the left corner. So if you imagine that this part in red is not visible, you would get a pattern that resembles a fringed perianth, for example, which increases the contrast against the background and potentially facilitates detection, flower detection.
Cool, thank you. Yeah, it's very, very interesting.
Yes, I also think so, but it's highly speculative for them.
Great, thanks.
Thank you.
Okay, we've got a question on the chat from Sergio Chozas. He says, I recently knew about Bombus terrestris stealing nectar. They are nectar robbers. So would it be an issue here? What do you think? Like using holes to steal nectar?
Yes, it's an interesting question, because this syndrome is particularly prone to cheaters. And in the field, you see that some birds, for example, pierce the nectar, the spurs, and rob nectar. But that's something that would be interesting to examine, because this high frequency of sphingophiles is apparently highest in the Afrotropics. So in South America, there are also a lot of examples, but not at the same level of Africa. And in South America, there are even some birds, clades that are specialized in nectar robbery. So eventually, nectar robbery pressure may be stronger in the neotropics. This is highly speculative. But yeah, so there's nectar robbery in these flowers. It's not extremely frequent, I would say. So most flowers you find in the field, they are not destroyed, but sometimes you really find birds like piercing the flowers. Some are attacked by herbivores, so sometimes you see grasshoppers eating the flowers, and then they are easily exposed to other animals to collect nectar. But I don't have data on nectar robbery.
Yeah, it's something that has been also seen in Linaria, which we know very well as well. With nectar spores, it was described in Central Europe, like this bombus that Sergio said. It seems they rob nectar from Linaria vulgaris, but it's not something that I have seen very frequently in wild populations here in the Iberian Peninsula. But it's an interesting topic.
Yes, clades that are specialized in nectar robbery and their possible impact. Yeah.
So, any other question? Sergio says obrigado.
De nada. Yes, feel free to ask questions in Spanish as well. It's just that I'm a passive user, but I speak Portuñol. Hablo muy bien Portuñol también.
So, have you looked at the effect of spores on diversification rates in ungrateful orchids in general?
I did. I'm going to show you some slides. So, basically, I calculated with colleagues from the Natural History Museum in Paris, Jérôme Colobert and Florent Marteau's. So we looked at diversification rates in angropoids, so only angropoids. And we checked, so here you have the diversigram, and here you have the variation of spur length color-coded with diversification rates. And we plot all these together. And you have here on the upper side the non-sphingophilous species, and the lower side the sphingophilous species. You can see that Some of the highest rates effectively occur in sphingophily species, but this is bimodal. But some of the, if you look at the diversigrams, some of the clades that show the highest diversification, namely Drumelia, namely Aerangis, namely Sertorchis, they are strictly sphingophile. But well, this is cherry picking. these are diverse and sphingophile, but this was not significant when we tested it statistically. But at least in some clades, they speciate very fast, and they are hawkmoth pollinated.
But all the species in this phylogeny have a spur.
Yes, all the except one have a spur. So there are 800 angracoid species. Only 6 do not have a spur.
Okay. So the idea here is that the lineages with the longer spurs usually or sometimes have the higher diversification rates, right?
Exactly. That's the case for Drumelia, for Sertorchis and Aerangis. Aerangis particularly fast and is hawkmoth pollinated. But well, this is not the general rule. There are also other clades that are well. Then basically what we have is that you have these clades with many species, sphingophiles, and then you have isolated lineages that are sphingophiles with only one species. So basically you have this pattern, either a lot of species or isolated lineages.
And who pollinates the other lineages that are not sphingophilos?
Sorry,
what are the pollinators of the lineages that are?
So we know very little about pollinators in angracoids. So most what we know is in hawkmoth pollinated species. So there's only a few cases, documented cases of pollination by other moths. So settling moths, phalonophily, in some green flowered long-tubed but not that long-tubed. And then in the Mascarene Islands in the Indian Ocean, we have evidence of bird pollination and even cricket, grass, orthopteran pollination, but well, in peculiar island environment. But so 30% of angracoids are sphingophilous based on the presence of long spurs. The other 70% are either mostly phalonophilous, so settling moth pollinated, and probably there are a lot of melitophilous species, those that have short clavate spurs, relatively bulbous short spurs. They are probably melitophilous. If you look at for example, the South American species, so there are only 2 genera of angracoids in the neotropics. One of them only has small, teeny tiny flowers, white with short spurs. And in this genus, the genus Campylocentrum, we've only found pollination by bees. And you find these type of spurs that are clavate, bulbous, short, also in Africa. We don't have evidence, observations, but they are probably pollinated by bees.
It reminds me a bit of the case of Aquilegia in America, with the different pollinator syndromes, including
the classical study.
Yes.
Yes. So this graph, as you know, this graph that I showed you comes from the study of Aquilegia, influential paper by Whittle and Hodges. And yes, so Aquilegia is really the most well-studied system. But these plants have really low visitation rate, and they grow on top of trees. So they are not the most user-friendly group for making observations. But now
you mean the orchids, the
epiphytic orchids, because now my colleagues are using cameras now to start recording pollinators. But it's still at its infancy.
Well, it took like a century to observe the actual interaction, right?
Yes, exactly. Really low visitation rates. Yes. For hawkmoths, because for other groups probably they're not that difficult to spot. It's just that they are not that showy. So we always look for the most extraordinary examples, the really exceptional species. But
Good. Are there any other questions? If not, maybe we can stop here.
Thank you. Yes.
So thank you, João, for this excellent talk, and
Thank
thank you
you.
everyone for attending. So see you all in like a month for the next session of this Cafés Sistemáticos. Thank you.